last week, we just learned hypersensitivity, right? So we learned hypersensitivity. So which one can tell me 4 types hypersensitivity? What's the name of the type 1? Type 1 hypersensitivity, also called immediate. Right. And type 2? Right. And type 3? Immune complex. Type 4? Yeah, delayed. Yes. So which type hypersensitivity is not involved in autoimmune disease? Type 1. Yeah. Type 1. No tissue damage, right? Okay. So today we will learn how the autoimmune disease can happen. Okay. So 3 questions for this lecture. Oh, wait. Here. Okay. So first question is what the difference between the autoimmune disease, autoimmunity and autoimmune disease? They are different. And the second are what are the 2 mechanisms of the autoimmune disease? And third is how can self-tolerance be breakdown in the human immunity? Right? Okay, we go to the first question. Is about what difference between autoimmune disease and autoimmunity. Okay, so we know that in the healthy individuals, so we can maintain a healthy state, right? Because the immune system works well. So 3 functions of the immune system. Can you remember? 3 functions of the immune system. Defense, surveillance, homeostasis. Right? Okay, so today we are talking about immune homeostasis. Okay, so we know that for the immune system, they need response, right? Before they need response, they need to recognize which is self, which is non-self. Okay, so for the healthy individuals, the immune system just recognize the non-self or foreign antigen. They cannot recognize self-antigen. If there is self-antigen, no response, right? If they are inventor or they have a non-self antigen, they have strong response. Is it? So this is a so-called, we call self-tolerance. So autoimmunity requires lots of self-tolerance. So this is self-tolerance. If there is self-antigen, no response. Is it clear? Okay. So what difference between the autoimmunity and autoimmune disease? So autoimmunity means everyone, our body can make antibody. So this antibody sometimes they make against our own tissues, our own cells, because this cell or tissue were damaged. It's not. It was died. All the cells died. So we need using the immune system to recognize these dead cells to clear them, right? So after clear them, they need to produce antibody. So this antibody we call what? Autoantibody, right? But this antibody can use to just monitor the autoimmunity. So autoimmunity is actually pretty common in most of healthy individuals, right? So autoimmunity, for everyone we have autoimmunity. We have the autoantibody in our body, but it's not disease. So when they have the disease, so if we can detect the autoantibody in our blood, in our serum, that not mean that we have autoimmune disease, right? So it doesn't mean that we have autoimmune disease. So when these antibodies, they start damage the tissue or damage the cells or organs, they will cause the functional or cause the physiological problem. We call it what? Yeah, autoimmune disease, right? So only some person, they have autoimmune disease, autoantibody or autoreactive T cell, and this T cell and antibody can damage the tissue of cells. They will lead to autoimmune disease. Is it clear? Okay. All right, so we go to the second question. The first question, we know that an autoantibody or autoreactive T cell, they are involving autoimmunity or autoimmune disease. Okay. And what are mechanisms of self-tolerance? We know that the autoimmune disease, only the self-tolerance is broken or is lost the self-tolerance, right? So what are 2 mechanisms of self-tolerance? Do you know? Mm-hmm. Self-tolerance. I think you have learned some in the previous lecture about the T cell and B cell development. Yeah? So they have 2 selection. Yeah. So which selection is involving the self-tolerance? negative and positive selection. Okay, so mechanism of self-tolerance including two mechanisms. One is central, second is peripheral. Why we divided the central and the peripheral? Because this tolerance mechanism happen in different sites, right? So if the self-tolerance just development in the thymus or in the bone marrow, this is the tissue, the T cell and the B cell development, right? So during the T cell, B cell development, they maturate to the maturate T cell and B cell. They will need to recognize self-antigen. If this cell can recognize self-antigen, this cell will die, right? So we call it negative selection. So it's happening in central immune organ, right? So we also call it central tolerance. So in the peripheral tolerance means this cell, the T cell, B cell, mature T cell, B cell, they leave the thymus or bone marrow, but they can recognize self-antigen, right? So in the peripheral immune organs, they will not react to self-antigen because they're using these three major mechanisms. The first is called immunologic ignorance. The second is called coronal energy. The third one is peripheral suppression T cell. Okay. All right. So what is the immunologic ignorance? So what is ignorance means? Ignorance, it means cannot see, right? Right? So it just occurs when the effector cell never meet the antigen because there are physical barrier between the T cell, B cell, or self-tissue. So ignorance, cannot see the antigen, right? And second is coronal energy. What does energy mean? Right, they have no response, right? Because we know that for the T cell activation, they need single 1, single 2, single 3, right? If there is only single 1, no single 2, or no single 3, no response. So this no response we call coronal energy. So coronal energy just occurs when the costimulatory molecules, they are not expressed on the most normal tissue. So normal tissue, there are no costimulatory molecule, so the T cell, they can recognize using the TCR to recognize the cell peptide. But because they have no costimulatory molecule, so no response. See? So this is called T cell coronal energy. The third one is called proliferative suppression by T cell. So we have learned that as a subset of T cells called regulatory T cell, right? So this T cell, they have the inhibitory function. They can inhibit the immune response. Right? If the T cell, the self-react T cell or B cell, they can use in the T cell, B cell to recognize self-antigen. But regulatory T cell, right here, they can inhibit this self-react T cell. Right? Okay, so this is the peripheral self-tolerance. Okay, just like here, this picture show here, and this is the central tolerance. And later generative lymphoid organ in the bone marrow and in the thymus, right? So this is the peripheral tolerance, so in the peripheral tissue, okay? So during the T cell, B cell development, they have precursor, and they will develop into the immature lymphocyte, and go to here is mature lymphocyte. After the maturation, they will leave the central tissue and go to the peripheral tissue, right? So in the central tissue, during the T cell, B cell development, if they can recognize self-antigen, they will die. And they will deletion. They will undergo apoptosis, okay? If they change the receptor, they cannot recognize the self-antigen, they will survive in the thymus or in bone marrow, okay? And here is during, if the T cell recognize self-antigen, and this T cell will develop into the regulatory T cell, inhibitory T cell, okay? And in the peripheral, we can see here, if the T cell, B cell maturation during the thymus or bone marrow, they will leave the central tissue and go into peripheral tissue. In the peripheral tissue, this self-react T cell, they can recognize self-antigen, right? Yes. But they can use in the coronal energy, they can use in the apoptosis, and using the regulatory T cell to inhibit the self-react T cell. So they still have no response. Okay? Okay, so this picture just shows in the thymus, in the immature T cell, they can recognize the self-antigen, but they will die. Okay? If the T cell can recognize self-antigen, if they are not dying, they will undergo development differentiation to the regulatory T cell. Okay, regulatory T cell. Okay, so just we can see here in the central system, not all self-reactive lymphocytes are detected during the T cell, B cell maturation, right? Because in the peripheral, the self-reactive can still be inhibited by the immune system using this ignorance, energy, and the suppression. Or some T cell, they recognize self-antigen, but they will die quickly. So it's also called T cell apoptosis, right? So in healthy individuals, the immune self-tolerance, so they can control the self-reactive T cell, right? But the breakdown of the self-tolerance, they will lead to activation of self-tolerant T cell, B cell. They will cause the tissue damage, and they will cause autoimmune disease. You see? Okay, so this is a peripheral. We have just to know that they can use if they recognize self-antigen, they will activate it, they will undergo apoptosis. Okay, so if they have recognized self-cell, but they have no second signal, it will cause energy, right? And if they recognize self-cells, but they have the regulatory T cell, they inhibit the T cell activation, so blocked in the activation. Only normal T cell, they are not recognize self-antigen, they recognize foreign antigen. They will have the effect. They will activate, and they will form the memory T cell, right? So this picture just summarize B cell. We're just talking about the T cell, right? And for the B cell, they have the similar mechanism. First is centrally, and second is the peripheral, right? In the centrally, the B cell, they recognize self-antigen, they will undergo apoptosis. Right? Only the non-self-reactive T cell, they can survive in the bone marrow. Okay? So some self-reactive B cell, they leave bone marrow, they will go outside. They can also use in the energy, using the deletion and regulatory T cell development, control the self-reactive B cell. Okay? They have similar mechanism to the T cell, just like T cell. Okay? So we have finished the second question. It's about what's the true mechanism of the self-tolerance. Okay? Now we go to the third question is how can self-tolerance be broken down? We know that in the healthy individuals, the self-tolerance is very important for us to control the reactive T cell, B cell. They are not killing the self-tissue or cells, right? But when the self-tolerance will be breakdown, okay, many factors. Okay, the first is the immune system. They cannot work well, right? For some person with the autoimmune disease, the immune system cannot work well. They cannot control the self-tolerance, right? And the second may be the environmental factor, just like the microbiological agent, right? And some pathogen enter our body will induce autoimmune disease. Okay. The third is maybe the genetic factor. Okay. So for the patient with the autoimmune disease, maybe these three factors combined together, they will cause autoimmune disease. Okay. So for the immune system, we know that for the healthy individuals, they have the tolerance, they have self-tolerance, they have proliferative tolerance, and central tolerance. So the immunological factor, including all these things, the first release of the sequestered antigen. So we should know what is sequestrated antigen. Okay. The second is the T cell energy will break down. Right? So the failure of the activation induces cell death. And this one is the failure of the T cell mediated suppression. And these three things, I think these three are the new things for you. It's a molecular mimicry, epitope spreading, and polychromos lymph node activation. Which is always not work well. Okay. So we now go to all these factors. The first one is called immunological factor. So failure of T cell mediated suppression. So we know that after the T cell activation, they will undergo activation-induced cell death, AICD. Do you learn before about AICD? No? It means we know that if they have the invader, they have the passenger antibody. So the T cell, B cell will recognize them. They will proliferation, right? They will differentiation. So maybe in our body only have 100 T cell, B cell. They can recognize special antigen, right? So they need proliferation. So maybe they have 1 million T cell, B cell. They can recognize the antigen. So after they recognize the antigen, they clear the pathogen, and all this 1 million T cell, B cell will go die, right? So how they can die? They can use this activation, induce cell death. So just like this picture shown here, this is the T cell. They recognize the antigen, right? They will proliferation of the T cell killing the target cell, and they will undergo apoptosis. So how they die? They express fast. They also express fast ligand. So using the fast ligands, the T cell will die. This means just like suicide, right? And this fast T cell, they also combine this fast can also bind to the fast ligand to another cell, right? So they will kill each other, right? So all these cells will die. So if the T cell activated, they express fast ligands, and they will die. So this die we call activation-induced cell death. Okay, for some patients with the autoimmune disease, this T cell they cannot die quickly. So all these cells they are not undergo death, undergo apoptosis, and this cell may be recognized self-antigen. Okay. And the second is breakdown of T energy. So we have learned what is energy means. So for the T cell, we know that they can using the TCR to recognize self cells, right? But because they have no co-stimulatory molecule or no self-tolerance, self cells, so there's no response. But for some patient, they with autoimmune disease, this self tissue, they can express co-stimulatory molecule. Okay, for the normal tissue cells, they are not expressed B7-1, B7-2. But for some patient, just like type 1 diabetes, for example, just like type 1 diabetes, the patient, the beta cell, they can express B7-1 and B7-2. So they can activate auto self-reactive cell. And this self-reactive cell recognize self cells and will killing them. And this cell will die. Okay? So the second mechanism is breakdown of T cell energy. Okay, just like B7-1, B7-2. And IL-12, they can induce Th1 T cell development. Okay? So high level of the self cells express the second signal or breakdown energy. Okay, so this is some upregulation of the B7 molecule. I think in the multiple sclerosis and rheumatoid arthritis. And I just mentioned that beta cell, all these cells they express B7 molecule. But for the individuals, healthy individuals, this tissue are not express B7 molecule. Is it clear? Okay. And release of sequestrated antigen. So what is means sequestrated antigen? Sequestered antigen. It means this cell is hide. This antigen or these cells are hide. So the immune system, the self-reactive cell, B cell, they cannot recognize the antigen, right? But for the patient, maybe they are caused by the pathogen infection. They will cause the hurt or other things. They will cause this sequestrated antigen release. Okay, so the good example for here is this disease. It's called sympathetic officer. Yeah. Do you know this disease? No? Okay, just like this picture show here, trauma, they will damage one eye. Okay, and this eyes antigen, they are broke by the structure. They cannot recognized by self T cell, B cell in the healthy individuals. that if they have a trauma, they will cause this antigen release. Okay, so this antigen released, and they were carried to the lymph node, and they will activate self T cell. And then the T cell activate T cell will leave lymph node, enter the damaged eye. They also go into the effect T cell. They also can go into the healthy eye. Right? So after the trauma, maybe the two eye will be damaged. Okay, so how can we do? As a doctor, if you are a doctor, you find a person, they have eye damage, and you know that they have sequestered antigen will go out, will release. They will activate T cell, and this T cell, self-react T cell, they will enter the eye through the bloodstream, right? They will damage this eye. They also can go into the healthy eye, right? So the good things we can do is to block the antigen release, or using antibody to block the antigen, okay, to help to save the healthy eye. Okay, all right. So this is a good question, good example for the release of sequestered antigen. And many autoimmune diseases, besides this one, multiple sclerosis, rheumatoid arthritis, and others, they also caused by the antigen release. Okay, right? And molecular mimicry. Do you know what this means, mimicry?
Yes.
Mm-hmm.
To copy
yeah, it's similar, right? It's copy, okay? So determinant, so we also call epitope. So determinant of infectious antigen, we know that in our body, the immune system can work well, right? So if they have the invader, they have the infectory antigen, this antigen enter our body, they can recognize by our immune system, right? But if this antigen, this infectious agent, they can mimic a host antigen. So that means that they trigger the self-react T cell, self-react B cell. This T cell or B cell can attack host tissue, right? So this T cell, B cell, they need to recognize the invader, because the invader mimic the host tissue, right? The epitope or the determinant, they are similar in the pathogen, similar between the pathogen and the self tissue. So after the self-react T cell, they recognize the pathogen. They also recognize the self tissue, so cause the self tissue damage. Okay, so example for the molecular mimicry, we can see this picture shown here. This is the self-react T cell. In the healthy individuals, they cannot be activated because they have no antigen, right? But if they have the pathogen, they have the microbe, the antibody. This microbe antigen, they are similar to the self-antigen. So they reactive self-react T cell. And this T cell recognize the microbe antigen, and at the same time, they will recognize self-tissue, right? They will cause tissue damage. And some disease, just like SLE. So in the patient with SLE, maybe the patient, they just infection by the EB virus. Okay, so because the EB virus, they have some antigen, they have some epitope that are similar with nuclear SM antigen. Okay, the second is rheumatoid heart disease. The antibody, they recognize the streptococcal M protein. This protein are similar to the cardioglycoprotein. So after the infection by these bacteria, they will cause the heart damage. Okay, I think there's a picture, just like here, the evidence. Rheumatic fever. So this pathogen, the person, they infected by the pathogen, they will cause sore throat, and the immune system recognize the pathogen. They will produce antibody or infect the T cell, B cell. And because this epitope on the bacteria are similar to the heart glycoprotein, right? So after the immune system respond to the invader, they also can recognize self tissue, so will cause cross reaction or cause the heart damage. So we call this acute rheumatic heart disease. Do you know this disease? Okay. All right. It's happened always in the children. See? Okay. So there are many proteins are similar between the microbial protein and autoantigen. I just using this table to show here. We can see this disease. Okay. EB virus, HSV, VSV, and HBV, tuberculosis, and other bacteria. Right? All these bacteria, all these virus, they have the similar protein to our tissue protein. Okay? All right. Okay, so another is called epitope spreading. What is epitope? Yeah, dominant protein in the antigen molecule, right? Or not antigen. So the epitope spreading, what is spreading means? Yeah, spreading means the antigen, the determinant can move from one type to another type. This means spreading, right? Okay, so the initial response to one self-determinant, one epitope, they could express to involve additional determinant on the cell molecule, right? So this means spreading. Okay, so we know that in one molecule there are many epitopes, right? Okay, so epitope spreading is help us to express how a response to the epitope they can mature to a full-blown autoimmune response of the response. Okay, so what cause autoimmune disease? We know that the major characteristic of the autoimmune disease are clinical or are persistent, right? If the doctor they can diagnose a person they have an autoimmune disease, that means the person they need to take drug all of rest of his life or her life, right? Because these diseases are persistent. Right? Why this disease can persistent? Because we know that we're using some drug to treat a person with autoimmune disease. They can control the self-reactive, right? Self-immune response, but the epitope will change, right? They will change to another one, so they maybe induce another immune response, right? A second time immune response. Okay, so the good example for the epitope, is it the epitope spreading in our body? Yes. So we can use in here in the initial response, maybe this epitope just the surface antigen, and after they damage the surface protein, they will cause the self T cell, B cell react to the anti-nuclear antigen. Right? Also, maybe in the nuclear, we know that histone protein covered the DNA, right? So in the initial response, they just have the response. They just using the self-react to the histone, but in the second time, maybe they can recognize DNA, right? So this picture just show how the antigen epitope happened, and we can see here. This is the T cell. They can recognize the X1. So they will produce X1, specific antigen, specific immunoglobulin G. And secondly, they can also recognize X2, right? And epitope spreading from the antigen X to antigen Y. Just like this picture show here, this is X2, the T cell. And they also recognize the Y-specific T cell here, right? So for the patient, as a doctor, if the patient, they take drug, they take the medicine, right? And maybe the inflammatory response will be controlled. But maybe 2 or 3 months, they will come here and just ask you, why I still have the clinical symptom again? You should explain why. Why? Yeah, epitope spreading. So you need to tell him or tell her, because you have the autoimmune disease, that means your self-reactive T cell, B cell can recognize the antigen. And this is the first time maybe they just surface antigen. The second time maybe they go to the nuclear antigen, right? So they will go on. So this disease may be persistent for a long time, right? Okay. All right. So polyclonal lymphocytic activation. So we know that during the T cell, B cell development, they need to express TCR, BCR, right? One TCR just recognize one type of antigen, right? But for the many pathogen, they can use in the super antigen. Do you learn before? Super antigen. What difference between the super antigen to the normal antigen?
I think the first one can activate the bigger response.
Yeah, they can activate a large population of T cell, right? So super antigen means they have the activity. The activity is they can recognize not just one type of antigen. They may recognize several antigen, right? So if the pathogen they can activate several T cell, several population of the T cell, B cell, and among this population, maybe some self-reactive T cell, B cell be activated. Is it clear? Okay. So here is an example for this is the bacterial endotoxin. They can induce mouse lymphocyte to produce anti-DNA, anti-cytomonocyte, anti-red blood cell, many different type of the antigen, right? Okay, so it's danger for the healthy individuals. Okay. All right. So we have finished all the immune system factors. So can you to know the sixth part of the immune system? The first one is release of sequestered antigen, and the second one is breakdown of energy, and third one, breakdown of the T cell, B cell apoptosis, right? AICD, and molecular mimicry. Energy we have got it.
Epitope
Spreading, and last one, Polychromos, right? So this is the sixth part of the immune system. They cannot work well, right? And now we go to the antigen. Break. Okay. All right.